What we want to talk about today, it's never too early to consider, for the considerations for gene therapy manufacturing in the early stages of development. As we all know, your phase 1, phase 2, if it's an unmet medical need or an orphan indication, can ultimately become some of your registration patients. So how do you think that through? So we have a great panel with us today. To my left, we got Fraser Wright, who is now a professor at Stanford. Formerly was at Accident, and formerly before that he was at Spark. So Fraser's one of our pioneers, so glad to have him on our panel. We have James Brown, Vice President of Corporate Development at Aldevron. Oh, I'm sorry. Michelle Berg. Sorry about that, guys.
Or James Brown.
James Brown. Michelle Berg.
The other James Brown.
President of Nucleic Acids at Aldevron. Susan Nichols, CEO of Falcon Therapeutics. Jeff Castelli, who is the Chief of Portfolio at Amicus. And Maritza McIntyre, who's the President of Advanced Therapies, LLC. So with that, I'm going to kind of read the problem statement, and then we'll let the panel start addressing some of the questions that we've prepared. And hopefully we can get some questions from the audience as well. So I'll just read the problem statement. So the success of gene therapy has resulted in significant benefits for patients and tremendous growth in the number of programs in development. Many programs have been granted accelerated designations, RMAT, et cetera, and have the potential to reach patients far faster than the traditional drug development timeline. Given the accelerated development and the complexity of gene therapy manufacturing, strategies to produce the therapy and manage the supply chain must be considered very early in the development process. We're going to discuss how the programs with limited time and resources can address manufacturing challenges and review some of the lessons learned from those in the field. So we're going to start off. First question is, how do you balance proof of concept with future needs with respect to phase 3 and commercialization? And Susan or Jeff, if you want to field that question.
I'll start, I guess. So I think I'm number 2. Good morning, everyone. So I'm Jeff Castelli. I'm the Chief Portfolio Officer, Head of Gene Therapy at Amicus Therapeutics. We got into gene therapy about a year and a half ago. We actually have 2 clinical stage gene therapy programs, AAV. Those are in rare diseases where there's a potential, we believe, to file a submission off of our initial clinical phase 1-2 data. And in those types of cases, it's really important to think about what's done early in the manufacturing process in order to enable you to potentially use that initial clinical material for submission. Even if you're not in that space and you have to do a phase 3 study, bridging from your phase 1-2 material to phase 3 is also going to be a big hurdle if you don't plan early for success. So I'll just mention a few quick things that we've kind of learned along the way that you need to think about early on. One simple one is just like which antibiotic resistance marker you use. Regulatory agencies do not want you to use things like ampicillin. You have to use things like kanamycin, and just being conscious of that because you'll have to change it later. Not the end of the world, but it's simpler if you don't and you think ahead early to what you're going to need ultimately. Making sure the cell line you're using early has traceability. Switching cell lines is a big change. If you're saying the same process, you might not need clinical comparability, but making sure early on you use a cell line that ultimately you could submit on is going to be important in terms of if it's a transient transfection hex cell line. I can't stress analytics enough. Having both measuring the titer and the potency are probably the 2 areas where people struggle the most. Current state of the art is ddPCR for titering. For your potency, early on companies just say, well, let's use an animal model in vivo release assay. Trying to get to an actual quantitative in vitro potency assay early will help you tremendously later as you're trying to bridge your material between different stages. Importantly related to that too is getting a good stability protocol, keeping retains of your material, showing that you have good stability long term, and also making sure you keep enough material that when you do change things, you can go back and test that material. Sometimes you're so excited to use all the material in the clinic that you don't keep enough to do good comparability testing later, and you don't want to be in that position. There's also lots of challenges around supply chain, which we'll get into later, but those are just some of the kind of early lessons we've seen about things to think about early on to position yourself for success down the road.
So some of the things I've heard when it comes to phase 1, phase 2 in gene therapy versus phase 3 is It just has to be GMP light. It has to be the spirit of GMP. That's really not what the FDA was trying to indicate. And Maritza, maybe I can hand that off to you. What's phase appropriate qualification validation as you move forward? And maybe we can dispel some of this GMP light concept.
Okay, so just starting there, because I was at FDA. I left in 2006, but I was part of the team that worked on that whole phase 1 GMP concept. And so you are correct that under the law, as in under the FD&C Act, all products have to comply with GMP. And when the regulations were changed such that a phase 1 material used for phase 1 did not have to comply with the GMPs and the regs, it was stated very clearly that you have to comply with GMP because that's the law. So the regulations were modified. But it also, and then there's an accompanying guidance that explains then what would be appropriate for it to be in compliance with GMP at phase 1. And so there's that. Then in terms of phase appropriate, I think that is an issue. That there is and early on, FDA is going to be looking more to ensure, for example, that any method that is intended to assure safety has to be validated, even for phase 1. The other things, they should be scientifically sound. But I'm sorry, I forgot your name.
Jeff.
As Jeff said, if your thought process is really that you have a potential home run, a potential one and done trial and program, then I often see this disconnect between this is what we're thinking in terms of the potential of our product, but in terms of our manufacturing development program, FDA says we only need to do this. Well, the two are, you're going to get into trouble later.
And I think it's almost the whole purpose of this discussion this morning.
Yeah.
So thank you. Next question, platforming and standardization of plasmid constructs, such as packaging and helper plasmids to accelerate development and streamline manufacturing. Maybe we can kick that off to you, Fraser.
Well, sure. Okay. Well, I guess my experience has been that doing, as Jeff had mentioned earlier, the concept of moving to something other than an ampicillin resistance early on is good. Quite often, it's not easy to do because there are pressing timelines, and to do the engineering and the reestablishment of a new plasmid, it takes time. But to do that quickly and early is important. It's an FDA expectation to move away from ampicillin resistance markers and the use of ampicillin in plasmid manufacturing. So that's an important thing to standardize quickly. I think there's a future question or a subsequent question here, but I'll just touch on it here. There are certain considerations in the plasmids, if you're using the transient transfection system, which I guess is a major theme here. And that is, certainly within the expression cassette, if it's an AAV, or I think this applies to a lenti as well, within the expression cassette, of course, this will be your product. And you want to make sure that everything within that expression cassette is what you want it to be. Not more, not less. Safety considerations, you don't have extra material. But also do keep in mind that there's a substantial amount of what are termed product-related impurities, which are small fragments of other bits of plasmids and other bits of DNA. So the consideration of trying to make sure that you have relatively innocuous features within all of your plasmid systems is an important consideration. You don't want nasty sequences somewhere in the backbone somewhere, because a small amount of that might end up as a product-related impurity. So standardization early is good. It takes time and effort. And it takes thought, being very thoughtful about exactly what would be the best. What would you like to say once you have a BLA ready to go? What would you like to be able to tell the FDA I have in my plasmids? Trying to think of that early. Michelle, I think you're on. I want to take this way. Yep.
Additional comment. So, of course, Aldevron sees ourselves as an enabler, a supporter of the field. And one of the ways that we've identified is to have those constructs that we have PLDX80, which is the UNC XX80. Helper plasmid, and that's been in the clinic numerous times. So what we've done is we have created ready-to-ship, off-the-shelf supplies that span from research grade into GMP to have that continuity, to have that consistency that I think what we're talking about will help enable that rapid trajectory, basically, from preclinical into clinical.
And it
commercial.
allows us to plan well also, because you start to look at your target product profile and work backwards. Okay, how much do I need? What grade do I need? If we have expedited approval, we also have expedited CMC and quality expectations. So understanding what they are and building that in from the beginning helps us to have a smoother pathway, because we have to fund that and resource it and be able to articulate it to investors also. Here is why we're building quality in from day one, because we want to accelerate the approval process.
Could I just also say, from a practical point of view, like, I've worked across a lot of programs, and like, when you see like this group and this group and this group all having to go and get their own, master cell bank of the helper plasmid, it drives you a little bananas, because you know that's just like done over and over iteratively, and if it's there and there's a unique, it's all been characterized, it just makes more sense for the field, I think, and for any individual developer. That's one less thing that you need to try to manage.
Saves time and money.
Yeah.
That's exactly the
Just
intention.
Building off that, this is a good time I want to just jump in on the supply chain, because it is really, with transient transfection and your typical 3 plasmids that you need to have, plus then your AAV manufacturing, we have 5 or 6 different programs we're trying to actively source plasmids for, and you need your cap rep, your helper, and your different transgene. Trying to plan ahead of time. Plasmids, you need to get your slot, you get to have your master cell bank made. That then needs to get released before you make your plasmid. The plasmid needs to get released. Now you need your AAV slot at your other CMO.
Yeah.
Getting all that to line up can take 6 to 9 months before you're even ready to get your other secured slot. So when you're doing that across multiple different plasmids, different suppliers, it can drive you bananas, but what can help is trying to use one helper. That takes one of your 3 plasmids out of the equation. The other thing is, thinking about purchasing some of these in bulk if you're using the same capsid across programs. Kind of store up your plasmids. It gives you a lot of flexibility later. As we're looking at which programs we're going to move forward on faster than others, potentially, and prioritizing, one of the things we've said is we just want to make sure we have plasmids ready to go no matter what in bank, because you don't want to have, all right, we're ready to pull the trigger on this program, move forward quickly. Oh, now we need to take 6 to 9 months to get our plasmid supply in place. So just thinking through that early and trying to standardize across your plasmids is key.
I would like to add to that, just as a shameless plug for all who provide plasmid in support of these clinical studies, that people don't think of the plasmid soon enough, and find themselves in just that position. And if I could leave you with that note, just please think of them earlier, because we can do some pretty amazing things. We can, really accelerate. We can do that, but it's very challenging when people come in screaming hot and realizing they needed it yesterday. So that's all.
Did any of you want to actually? One of the things you just touched on was the power of platforming, and starting with plasmid. So really, the two variables are the transgene, and maybe you might want to modify for tropism. Maybe one of you, maybe Suzanne, do you want to field that question?
That's not my area of expertise. I just
Maybe
looked at
Fraser. Why don't Fraser take that?
Okay, so platforming and tropism. So if I understand correctly,
The
Greg.
question is,
Yeah.
a lot of your plasmids are going to be common, regardless of indication.
Right.
The 2 criticalities are, what is the transgene you want to
Yeah,
express?
and tropism.
And then
Yeah.
tropism.
Understood, right. Well, I think that is, so I think those, aside from the adenoviral helper plasmid, which is standardized, and I think this is, to standardize that early, it's used for all products, AV products, let's put it that way. You're right, that's true. You have what I would call the vector plasmid, the plasmid that expresses the transgene, and that has to be defined individually. There are certain considerations for quality around that, which I thought might come in a subsequent discussion. But that is kind of a case-by-case plasmid considerations with certain principles that have to be applied. And then I think in the 3 plasmid system anyway, which is probably the most common, I would call this the packaging plasmid, the rep cap. And of course, cap encodes the serotype for your vector. And if it's a CNS indication, you may want to standardize on 1 type of serotype, because it consistently gives you a certain biodistribution and delivery. For a systemic administration, you may want to standardize on others. So that's kind of intermediate. I think the transgene plasmid is customized per project, per indication. The vector plasmid, or pardon me, the packaging plasmid, well, it's kind of for systems of administration, and that may be partially platformed to a degree. And then, of course, the adenoviral pepper plasmid is standard across the board. So that's an attempt, I think, to.
Greg, I would just add to that, when you have multiple gene therapy AAV products, some are going to have the same capsid, some might not. It's not only the benefit if you have the same capsid for multiple programs. Now you can purchase your rep cap in bulk and your helper in bulk, hopefully, and only need to modify the transgene.
Yeah.
But even as you get to your AAV manufacturing process, if you've established with a certain capsid how to make your AAV and that's all done, when you switch just out the transgene, that's a much smaller change, and you can pretty much quickly get into engineering and production versus having to go through all the early stage process scale-up. So there's a lot of synergy in sticking with the capsid, but that being said, you're ultimately going to want the best capsid for that disease indication. Right. So it's trying to balance those factors. But it's not just on the plasmid side where if you stick with the same capsid, you get a lot of synergies in the later stage AAV manufacturing as well.
But I think there are some groups then who are, for example, focusing on neurological, then that pretty much, they pick a plasmid and then they have a
Serotype.
variety of a serotype, and they have a variety of indications.
Yeah, and I think that's huge, because really to fully characterize biodistribution and efficiency of going to the right cells is an enormous endeavor.
Yeah.
Pre-product development, this is an enormous endeavor. So to really say I understand well the biodistribution could be applicable for 3 diseases. Right. To platform in that regard is very helpful. You do want to get the right biodistribution, of course, too. So making the right decision at the beginning, to the degree possible, is important.
So we focus a lot on plasmid, but there are other considerations. The next question on the table will be, what are the considerations with respect to other raw materials beyond plasmids? I don't know if you want to handle that, or someone else in the panel want to grab that question. So, yeah.
Well, yeah, so for example, if you go to lentiviruses for ex vivo gene therapy, then that actually becomes a critical raw material, or maybe it's the drug substance. Kind of depends on how the regulators view that. But then, obviously, then depending on your lenti manufacturing method, then you could have layers where you've got your plasmid and your lenti before you start to produce your product. So. That's a complexity. The producer cell itself, and then if you're adherent, you have serum. If you're using affinity columns, then there's, you have to characterize leachables, et cetera. So there's a lot of other, critical raw materials that go into the whole process.
Fraser, you look nodding your head.
Yeah, I'm nodding my head. Well, and synergizing really with what Maritza has said, having been through the process through a licensure, I think some of the things I'd say is there's always consideration, just like the vaccine or biologics industry in general, can we replace animal-derived components with other components? That's always a good thing. I don't think it's a necessary thing to do, but it's always a good idea. Examples, of course, are serum, for production going to a serum-free system, if you can do it reasonably. There are timeline considerations, et cetera, and process change considerations. It is preferred if you can move that way, certainly. Another example is trypsin. The use of trypsin, if you're using adherent systems, not using porcine-derived trypsin, using a recombinant version. These are, I think, fairly obvious examples, and I think they're probably reflective of what the biologics field in general goes through in assessing raw materials for GMP and commercial level products.
Fraser, just quickly to add, I know we do also want to get to questions, James. Depending if you're in a serum system, which a lot of people are early on, different CDMOs actually, some will supply the serum for you, and others will actually want the sponsor to supply serum. So that's something to be aware of as you're having those discussions. You don't want to get surprised at the end of the day, be like, oh my God, now we need to source the serum. So just something to be aware of, to put on your checklist as you're going through on how you're going to do all the different supply chains.
Yeah, and serum is not, of course, this is stating the obvious, not picking up the catalog and saying, well, that's a good price. It might more involve an audit visit to Australia and things like this. Obviously, a huge consideration if you are using serum, selecting an appropriately qualified reagent.
Well, but actually, to expand on that, Jeff, is that it's the same with the cell bank. Like some CDMOs they have a cell bank that they will use to manufacture your product, but a lot of them don't. And so then that is an extra step that you have to actually do, is to establish and qualify a cell bank.
With extremely long-term consequences.
Yes.
It's like getting married.
Yeah. And
Or
not,
having children, or having children.
yeah, so that's a huge one right there, yeah.
Let me shift gears a little bit. So a lot of folks are getting into the clinical trials using quote-unquote 2D. Us older guys call it adherent cell lines. And then they want to scale up and put it into what they call 3D, and that's what us older guys call suspension culture. So, so getting into phase 1 using hyperstacks or cell factories, but ultimately moving into stirred tank reactors or potentially the iCell packed bed. And so there's going to be a shift. There's going to be a need for comparability. So let me hand that off to the panel, and which one of you would like to field that question? Thinking about the comparability from anchorage to suspension. Go ahead, Fraser.
I can start a little bit, and I've had experience in both areas. I think it is interesting to see that the products that we have licensed in the cell and gene therapy space, there's a substantial, I don't want to say exactly how much, but component of adherent cell culture for production. I think it's just a fact. So I think what people are faced with typically are that these programs emerge, maybe from academia, with adherent systems. And people are moving forward quickly. If this is a viable product, if it's a promising product, it shows its promise early, and there's a great acceleration to move forward. So a transition from an adherent system that is working in the preclinical, the tox study, the phase 1-2, to a more scalable suspension system, in principle, is a great idea, but it is challenging in the context of rapid product development, especially where the CMC components are probably on the critical path because of the nature of gene therapy. So I'm not saying it's, of course, we should all be aiming for better suspension systems, high productivity, serum-free systems, but I think the key component there is the comparability studies, planning prospectively the comparability studies that would be required to go from one production manufacturing process to a more advanced commercial level manufacturing process. If you're going to do it, plan it early, figure out what you need, and have qualified and appropriate assays in good shape, really, ideally very mature shape, so that you can change with confidence. Assays that do really measure the critical quality attributes, the most important quality attributes of the product as you go from process A to process B. So a couple of comments.
Yeah, no, I think that's really key. And I know that, as you said, the reason why, most of these approvals are adherent is because that's what they were. But then from my point of view, and of course I'm not there doing the day-to-day, but I see a lot of pain trying to actually scale and qualify that adherent assay. And when I ask, well, why didn't you just switch to suspension? They say, well, because we couldn't. And I wonder, my point of view, without actually being there doing the dirty work, is that you should be able to do it if you have assays. If you have good assays, and if during early phase development you weren't just doing the minimal characterization that was required for release. Because then you would have a body of information that then when you went to your much more scalable and easy to validate suspension system, you could do those comparisons.
Exactly. I mean, AAV is a biologics.
Yeah.
In biologics, we change processes,
Yeah.
we show comparability, but we have good analytics.
Yes.
I think that the key is having those analytics, and where a lot of companies have struggled is not having good analytics early on, changing processes later, which comes back to the importance of early on figuring out what is your commercial market size. Is it a rare disease, big disease? Are you going CNS delivery into the intrathecally, or are you going systemically? Because if you're in a rare disease and it's CNS delivered, adherent cell culture is commercially viable, and you're in a great position because you might even be okay in hyperstacks. Preferably you get to maybe something like iCell, which is a little more scalable. But that's one path versus knowing we're in a larger rare disease or not rare disease, and it's systemic high dosing. You're going to need to be in suspension, transient transfection, and that might not even get you there. Companies are starting to look at co-infection or stable cell line. But ultimately, clinical proof of concept data is invaluable early on. So if you get in there with your adherent material and you want to switch to that larger scale suspension or co-infection or stable cell line, make sure you have good analytics to allow you to do that comparability and bridging. It's going to be key.
And again, just to, I guess, triple emphasize assays.
Yes.
So it's not easy. The problem with the gene therapies is it's very challenging. It's not that we don't have a lot of good brains out there trying to figure out how to do a good assay. It's easy to say, do good assays early. The problem is it's very, very challenging. And as just a couple of examples on this, potency assays, which are very important, a critical quality attribute is, of course, the specific potency of a product, are very challenging for gene therapy products. There are guidances from the FDA, but they're just nevertheless very challenging. Do not underestimate what it would take in terms of millions of dollars and time to fully develop and validate a good potency assay for a gene therapy product. And also to go back to, I don't want to call it a trivial assay, but folks might think that just trying to quantify the titer or the concentration of a given gene therapy lot is, well, that should be pretty basic. We all do it. I think with monoclonal antibodies, it's an OD assay, I believe. I'm not sure. I haven't worked in that field. And it's pretty precise and accurate. This has been a huge challenge in AAV vectors and also for lentiviral vectors to get even within a half log precision. I think in the earlier stages of development, it's a threefold precision levels. Accuracy aside, the FDA has convened workshops for this, trying to get all the players in the field. How can we better standardize our titering assays? So, it is important to get the assays right. How we get the assays right are a big challenge, so it's important to put a lot of resources and smart resources into the assay development early to support comparability for process changes, et cetera. So I think just to sort of build a little bit in the comments.
Are there any questions let me take a pause. Any questions from the audience that we can field before I continue to ask questions? Okay. All right. So one of the things you brought up, Jeff, was basically the target product profile. And my experience, even in phase 1, it's good to at least have a draft target product profile and kind of what is the indication you're going after, what is the potential population, because that's going to help drive development. If it's a small indication like Tay-Sachs, why would you go to a 200-liter stirred tank reactor? If it's Parkinson's disease and you're going to have 20, 30,000 patients a year, it's a whole different ballgame. So maybe you could comment on how you might want to start thinking target product profile early on.
Yeah, no, I think as I mentioned earlier, just figuring out what is your ultimate patient population size, and is it delivered CNS delivery where it's usually an E13, E14 kind of dose, but not per weight, versus systemic where it's that same dose but per kilogram, and so you need 70 times more of it. You can run those numbers and pretty quickly know whether something like iCELLis is commercially viable. Generally, when we look at the numbers currently, and there's ways to bring down costs of transient transfections with plasmid supply, and I don't want to say this with some of the plasmid suppliers, but some companies like Pfizer are looking at sourcing their own plasmids as one way to kind of drive the cost down to make something like transient transfection suspension commercially viable for these larger systemic indications. But there certainly is, as we look for diseases that are larger and systemic dosing of AAVs, there's still a huge gap right now in terms of what we can make and the scale we can make it at versus what's going to be needed. And I think as we start to go more towards biologics type manufacturing of suspension and larger reactors and maybe perfusion, there's tons we can learn from what we've done on antibodies and then other biologics. But we are just so early on in the AAV, at least manufacturing, as well as I assume lentivirus and ex vivo. But putting a mind towards that so that you can start to plan early for how you want to get there is important. But sometimes it's too big of a bridge to say, oh, we need to get into the clinic. Let's go with our co-infection process. Like that could take you 2 years to figure out before you can get into any and know whether your product actually has a clinical effect. So I think there's going to be a need to do some bridging and changing processes. Even though I know FDA is coming out and saying, we prefer you stick with the same process, I think practically that's just not going to be viable for most companies at some point in time.
Yeah, so 2 points on that. If the company's well-heeled enough, such as Big Pharma, they can run what I call the double drill. They can get into phase 1, phase 2 using an adherent process while they're developing their suspension processes. So then to demystify cell and gene therapy a little bit. So you hear about HEK293 adherent cell lines, and companies are trying to have breakthroughs and to go into suspension culture. So having been back at Eli Lilly for Zygris, we were able to get up to 3,000-liter bioreactors at 2 times 10 to the 8th, not 7th or 6th, 2 times 10 to the 8th cells per mil in perfusion culture with HEK293. So So it is not a big leap to get into suspension culture with HEKs. I predict the future is going to be HEK 293 in suspension. Producer cell lines will be the future of the gene therapy. And some of the folks in the audience are some of the leaders in making in the technology around producer cell lines. So my prediction is, and I think it's a safe one, that will be the future of making viral vectors. Yeah, that process was a 180,000 liter process over a 30-day period, so it's quite a process. So maybe we can talk about that a little bit. So producer cell lines will be the future. I don't know if somebody in the panel would like to field that.
I'll put in my 2 cents worth. I believe, the reason that we are using transient systems for AAV and lentiviral vectors right now, and these transient systems, of course, are transient transfection, but also recombinant baculoviruses and recombinant herpes simplex viruses, is because of the cytostatic, cytotoxic nature of some of the genes that are required to, assemble these things biologically. So we can't sort of build them in to a cell line and just turn it on. It's very hard, even though this has been the standard, I think, in the field for biologics in general. So I think as companies are maturing inducible promoters that you can have all the components in a cell line stably transfected, turn it on when you've got the right cell density and get this enormous amount of vector made. I mean, that is aspirationally, I think, the best way to go when we can, but it's not an easy thing to do. People have been working on true production cell lines for many years in the AV field. I know that at least 20 years, and our technologies are maturing substantially, and I think we have a better shot on goal in this, but it's not an easy thing to do. But yeah, ultimately, it's going to take away a lot of the supply chain constraints. Aldevron will be evolving their business model, I'm sure, appropriately. This is a multi-year, multi-decade type of prospect, I think.
I think so. That has been achieved. There's some for lentiviruses.
That's true.
And actually, in FDA's most recent CMC guidance, they talk about how that using that system reduces the risk of developing RCL. But the other part of it, though, is that so on the one hand, we're making a lot of bets, which are reasonable bets that you're gonna score on your first with your first clinical candidate. But people like Fraser and I, who've been in this field for a long time, have seen
more than 10 years.
have seen the morphing of even for factor 9, it's gone through lots of iterations. But once you commit to your producer line, that's like a marriage. So usually most people are not going to start at a producer's line because you don't know what your efficacy is going to be or whatever.
I mean, just to add on, if it's okay, a clear advantage of the transient transfection early on is that you can really explore different constructs, different serotypes, and that can go through preclinical and even phase 1, too. Yeah. It's very powerful because you have a lot of breadth. I mean, the true benefit of a producer cell line when we can get efficient ones going, and I think we're getting there, is that you can make in a much more cost-efficient manner a single product at large scale. So those are things that come into play weighting-wise, at different stages of product development.
So one thing that plays into our discussion today, and I'm just going to throw this one in, I know it's going to be fielded later during this week, is cost of goods. So right now, Fraser knows he was from Spark, was 400,000 an eye, I think, something along those lines.
425. Yeah, I think that's the approved.
I was at Novartis for Kim Ryan. I think we were at 750,000 for a course of therapy.
But 2 eyes came up at 850. Just further right. Okay.
So the point is, these are the first entrants into the field of gene therapy where the insurance companies and the government are willing to reimburse. But as more and more gene therapies come onto the market, it will not be sustainable to have those costs of therapy. So one of the drivers of the high cost of these therapies is the cost of goods. How much does it cost to actually manufacture a dose? So it plays into target product profiles. It plays into producer cell lines and suspension culture. Back in the day, I'm old enough to remember when monoclonals came along and we can only make 50 milligrams per liter. Now we're making 10 grams per liter. So there's going to have those kind of breakthroughs. But again, initially, high cost of goods was tolerable. And now if you look at the biologics field and the vaccines field, those costs have been driven down almost to commodity costs of goods. So at some point, for cell and gene therapy to be sustainable and hit multiple indications and that we can actually afford with Medicare and insurance companies, et cetera, we're going to have to bring it down. So I just wondered if anybody in the panel has any thoughts along cost of goods and what your thinking might be.
I mean, I would just agree with what you said, Greg. It's ultimately going to be important to get your cost of goods down. In rare diseases, generally where the pricing are, you see what's the first few gene therapies that have been out there, pricing in that $1 to $1.5 million per patient range. With that, you still have challenges with cost of goods if, it's a systemic administered product, for example. So, and you're in an adherent type system. So to me, it also goes along with being able to supply the market, because cost of goods is one bit. But when you run the numbers in terms of if you're at a systemic dose, relatively large patient population, and you just look at the number of Icelis 500 runs you would need, for example, you're like, you can't do it. There's not enough space, and practically, obviously that's tied to cost of goods as well, but you want to consider both, can we actually supply the market, which to us is the most important, and then cost of goods too, making sure you can at least, get some money back as well. But both kind of tie together, and I think ultimately, Greg, as you said, going towards something like a stable cell line approach with some of the great new technologies on inducible promoters, which will be great to see as we have proof of concept across different capsids to see that that really is working, is one of the good potential options out there. But even just getting into suspension transient transfection, I think seems like a lot of the early manufacturing is still mired in the adherent systems. And I think we're going to see that change. I think more and more companies will start to go into suspension earlier, which at least gets you into that mid-range. And then whether, what your ultimate solution is will be something else.
Go ahead, Fraser.
Yeah, so just to make a couple of comments too. So, I would like to, and I think it might be stating the obvious, but I'm a numbers guy, so I like to go back to numbers. So it is important to recognize that doses for gene therapies that are out there right now in development and licensed range over an enormous range. Low end is 10 to the 11th, and what does that mean? In billions or trillions or whatever. And I think the high end per dose might be as high as, well, it's certainly in the E16s. So we were talking about a range, and this is per patient, this type of thing. So that's a range that spans, 100,000, maybe a million. So you do have to keep in mind that the cost of goods to make this stuff, per unit is going to be similar, depending on the efficiency of your manufacturing process. But, the demands range enormously depending on your indication. And I think the eye indications are at the low end. And I think things like Duchenne's muscular dystrophy are way up at that high end. So that's one consideration, just to put on the table to understand. So cost of goods may be a minimal component, not a minimal, but much less component when you're talking about a low dose indication, and the overriding component when you're talking about a high dose indication. I'd also like to just touch on, again, numbers, productivity, specific productivity. Greg, you mentioned specific productivity. I think of that, grams per liter, whatever it is, as specific productivity. I think the specific productivity for the viral vectors is still variable and lowish. And we need to sort of work in the upstream processes to try to get those numbers higher. And I would just put out there that probably making 1 million AAV particles, I don't want to talk about lentia, 1 million AAV particles, 10 to the 6 AAV particles per cell in some system is a reasonable target. I think those are the numbers that are achievable. 10 million I don't think is achievable. Probably 100,000 is more typically what people are dealing with right now. I'm not sure, I don't know. But 1 million is a reasonable number to achieve. And then you can sort of back calculate from there. How many cells do you need? What's the density? How much volume do you need? As long as you can maintain that specific per cell productivity. So just to put a number out there, it's a reach number, it's an aspirational number, but those are numbers that really correspond to feasible biological high productivity for AAV vectors.
We only got a few minutes left. And one thing we didn't touch on was drug product or route of administration. So, Fraser and I worked on Parkinson's disease. It was relatively invasive where you actually had to drill holes into the patient's skull and put in cannulas to introduce the lentiviral vector into the brain. Then you look down at University of Penn, people like Jim Wilson, who are now have figured out how to get AAV across the blood-brain barrier and have dramatically improved tropism. So with respect to, and then you have to look at comparability of your viral vector with your delivery device. you could wind up with nonspecific absorption and other issues that are just typical. If you're thinking through a biologic, you would have to think through that. And I think that type of thinking now needs to be introduced into viral vectors. But route of administration and drug product, does the panel have any thoughts along that? We just got a few minutes left. I thought maybe we could close on that. We got 3 minutes.
Well, I always have an opinion. I mean, go for it, okay.
Go ahead, Fred.
Well, okay, so I think it's a good thing to understand that systemic administration to transduce CNS tissues, I mean, it's obviously a much easier route of administration, but the truth of the matter is that the doses required are markedly higher. Because if you do put any AAV, I think that we know about right now, and some folks may argue in the field, most of it goes to the liver. If you put it into the blood, most of it goes to the liver. It is agree that certain serotypes, such as AV9, I think is the one that's out there that people know, and maybe some other variants, do have a substantial ability to cross blood-brain barrier, so you can actually get CNS-level transduction following an IV administration. And I think this is the basis for the third, is it the licensed product coming out of Avexis. I believe that that's a CNS or a systemic administration. So just do keep in mind that, you know, tropism is important. Route of administration, parenchymal or a more direct route of administration probably will correlate with less exposure to the immune system and lower dose, you know, to get the same effect if it's not, by definition, a systemic requirement. So to go directly into brain tissue will probably require a lower dose and get it more specifically to an area, but of course it's much more invasive. So I think it's kind of one of these pros and cons, and you have to do an assessment of the risk, benefit, cost, you know, for any route of administration.
James, did you have a question?
You know, we don't have a lot of time, but I was going to ask about the non-viral stuff.
Yeah, that's one last question. We got 2 minutes left. Let me just field that real quick. So there's a lot of non-viral out there. There's now the cationic nanoparticles, et cetera. So non-viral delivery of the transgene to the target cells. I don't know, Fraser, you're nodding your head. Do you have some thoughts?
I always do, but does anybody, I mean, I don't want to keep them. I do have a strong, well, not as strong, but I have an opinion.
Right now, we're busy with AAV, but yes, we were looking at other approaches, and certainly getting away from the viral system would be great, but when you get the right tropism, and you know, there's lots of potential advantages, but
the holy grail.
Maybe I can bounce off from Maritza a little bit, and maybe I'll put my perspective on and see, because you've had a very broad exposure, if you can talk about it, to different systems. I mean, I've always viewed the viral vectors as incredibly efficient because they have evolved over the millennia, the eons, or whatever it is, to efficiently deliver DNA in a relatively stable manner, and that's why we use them. You know, they're complicated to make, but they're incredibly efficient. So I've always thought that the non-viral systems, it's going to be a lot of work to try to truly engineer a nanoparticle, you know, a synthetic nanoparticle that has all those efficiencies. But, you know, I haven't really been involved with the non-viral system, so I'm not sure if, Maritza, you'd agree or disagree.
No, I agree with you. I think, obviously, I mean, there's been work on that, so in the whole mRNA and
Oh, yeah, that's
so
true.
that I think will now cross-pollinate. But then there's, especially if you're talking about AAV, there's also the genome and persistence.
Yeah,
so
long term.
Yeah,
Yep.
that's also something that AAV has evolved. So it will come. I don't know that it'll come that quickly. And those non-viral particles also have their own toxicity profiles that have to be explored. So.
Thanks.
Well, we're out of time. I want to be respectful of the next group that's coming up, so it's 8 o'clock. I'd like to thank the audience for listening to us. I'd like to thank the panel for all their contributions.